Hi, my name is Mr. Torres, and today I would like to talk to you about my summer research with the RET program of 2013. So the name of my project is Computational Model of Functional Hyperemia. So you're probably wondering what that means. It simply means that there's an increased blood flow to certain regions of the brain where it's active. In other words, when neurons talk, usually one neuron releases a neurotransmitter such as glutamate that is sent across a synapse to affect another neuron. And in doing so, it basically burns up oxygen and energy. To replenish this oxygen energy, the local blood vessels has to dilate so that more oxygen and more energy can be delivered to those tissues. This is a really important function because if this is not regulated, even for a few seconds, it could cause a person to lose consciousness. And if this occurs for a prolonged period of time, let's say 2 minutes even, it could cause permanent brain damage and even death. And so here's an image of a brain using a technology called bold fMRI, or blood oxygen level determining functional magnetic resonance imaging. So this technology allows us to basically view the brain and see what areas of the brain where there's a lot of oxygen being delivered, which also then implies that there's an increased blood flow to that region. So to get a better idea of how this technology works, let me show you a quick video.
Simply put, the magnet tracks hemoglobin, the iron-rich protein that makes your blood red. When hemoglobin transfers oxygen to your tissues, a tiny change occurs in the hemoglobin's magnetic field. So basically, when your brain cells call for more oxygen, this big brother can see it.
Okay, so to show you another way, to explain to you another way what's happening here. So let's say neural activity is occurring, and usually there's a delay of 1 to 2 seconds where the fMRI response, in other words, the amount of oxygen has been delivered to certain parts of the brain. There's a delay here, again, 1 to 2 seconds, and then reaches its maximum at 5 minutes and usually reaches the baseline at 10th minute. So this is typically what you see during functional hyperemia. And so this is also called the hemodynamic response. So you're probably wondering, well, how does neuroactivity cause change in vasotone? So to explain that, let's look at this model. So what this model is showing you basically is, let's say this is a cross-section of your brain, this is the artery, and this is your vein. The artery, as you can see, digs kind of into the brain tissue where it basically bifurcates, or in other words, divides into many different smaller segments called arteries until it becomes capillaries. As you can see here, artery is surrounded by this peel coat, which is a membrane that separates basically the blood and the brain tissue. And so let's say that these dots are neurons. And so question is, how are the information of neuroactivity from these neurons basically being transferred all the way across to these arteries so that it knows that it needs to dilate? So that's really the hard question to answer for some time. For centuries, we've known about this phenomenon, but it was hard to explain. And to explain that, let's look at this model here. So this is a model that I made to explain what's going on within the brain. So let's say this is a peel arterial, and this is a smooth muscle that surrounds that smooth muscle here. And so let's say this is a peel coat, and this is your neuron. So let's zoom in here. So what we're seeing here is neurons talking, and there's this overflow of neurotransmitters such as glutamate. And so what we didn't know was that there are certain type of glia cells called ascites cells, which are really important in basically detecting what's happening with neurons, and then they could transfer the information all the way across to where there are smooth muscle cells. So there's this protoplasm ascite, which is located in the brain region, which can detect this neural talk here that's going on. And so basically it could detect the neurotransmitter, and that information is relayed across astrocyte and then to the next astrocyte. The way we could track this basically is that when astrocytes pick up information from a neuron, such as glutamate, it's going to release calcium. And calcium channel is going to be released, and then calcium basically going to travel from one end to the other. And so this is how the scientists, are able to track what's going on there. So let me show you a quick video of how that works. So here's a video of astrocyte cells in a petri dish. The the lighting of the exercise here indicate that the calcium is being released, and this right here, what you're seeing is calcium propagation being sent. Okay, so this is how we're able to track how exercises are signaling one another. To clarify a little bit, when ascites are similar to each other, again, calcium travels through ascite, but then the gap here is actually releasing glutamate across here. Okay, these little gaps. Um, and this last ascite, which is called prevascular ascites, that are interfacing with smooth muscle cells, they're releasing certain type of vasodilators or constrictor depending on what type of message it got from the neuron. So in our case, we're talking about glutamate, so basically a vasodilator, which is going to cause smooth muscles, basically to relax so they could dilate. Okay. All right. So then now that we kind of know all that stuff, I had to develop a computational model of functional hyperemia, and I had to use a math lab program. And at this point, I just want to thank Ian for helping me out with this because much as I love math lab, I think it's amazing. I did not know how to work it very well, but thanks to Ian, I was able to get a lot of work done with it. So the objective for my model here was to see, first of all, the transference of glutamate concentration from neuron to ascite to smooth muscle cell. And then the second part is really it really is the key point of this whole research, basically, is to see how this transference of glutamate to smooth muscle cells causes vasodilation. Um, So to basically have this model working, I need to know the key components. So we need to know about neurons, astrocyte cells, and smooth muscle cells, and basically how fast are these signals being sent, which basically is indicated here, and then the time it takes, which is again 1.2 seconds roughly, plus or minus 0.6 seconds. So there's a range there, and then based on that, I was able to determine the rough estimate regarding the length. So with all that information, I plugged it into the MATLAB program, and used differential equation, things like that, to basically determine the rate in which transference of glutamate concentration is occurring. So let me show you the data. So the data here, this first graph is indicating transference of glutamate concentration. So we have your neuron, we have your astrocyte cells, and smooth muscle cells. So neuron, basically starting with about 1,000 micromoles of glutamate, is releasing that glutamate, and let's say this is sending it to astrocyte cells. Astrocyte cells here in green is basically picking that up, but then also releasing it because it's sending that glutamate to smooth muscle cells. As you can see here, smooth muscle cells is picking that information up and reaches its peak at 5 minutes. 5 seconds, correction, 5 seconds, sorry about that. Okay, so the next slide here is showing you the change in vasodilation as the smooth muscle cells are receiving the glutamate concentration. So what you're seeing here basically is that at roughly around 0.4 seconds, it's starting to dilate and reaches its peak around 4.5 seconds, which is very similar to the hemodynamic response, as you can see here in this graph, as we talked about before. So the model looked very promising. And at this point, I just want to say thank you to, again, Ian for helping me out with everything, and Dr. Lerninger, and everybody else in this program. I really enjoyed it, and I had learned a lot. Thank you very much.